Today, we're diving into the fascinating world of the Automated Complete Blood Count, or CBC. For a lot of clinicians and laboratory professionals, the hematology analyzer feels a bit like a mysterious black box. Blood goes in, numbers come out. But today, we're popping the hood. We're going to transform how you look at that box, because it's not just magic. It is a highly sophisticated, multidimensional particle counter. Let's get into it.
Here's our roadmap for this explainer. We'll hit core technologies, RBCs and histograms, platelets and reticulocytes, WBC differentials, errors and flags, and wrap it all up with the workflow.
Section 1, Core Technologies and Evolution. It's wild to think about how we got here. We literally went from painstakingly counting cells by eye on a glass hemocytometer in the early 1900s to the absolute game changer of electrical impedance in the 50s. And today, it's like sci-fi. Robotics handle everything, automated sampling, barcode reading, mixing, even making the slides. Meanwhile, we're using multidimensional fluorescence and digital imaging to physically interrogate millions of individual cells every single second. It is a staggering leap in technology.
And it all really starts here with the Coulter principle, or electrical impedance. Think of the analyzer like a bouncer at a very exclusive club. The blood gets diluted in a conductive fluid, like isotonic saline, and then the cells are forced through a tiny doorway, an aperture, one by one. Now, cells are terrible conductors compared to the saline. So every time a cell squeezes through, it displaces the fluid and creates a little spike in electrical resistance. Two huge things happen here. The total number of pulses gives us our cell count, and the amplitude, or the height of the pulse, tells the machine exactly how big that cell is. Tiny pulse, you've got a platelet. Larger pulse, red blood cell. Simple, but brilliantly effective.
But we don't just stop at electricity anymore. Modern analyzers also use optics. The cells flow through a flow cell and get hydrodynamically focused. That's just a fancy way of saying fluid dynamics force them into a neat, single-file line, then bam, a narrow laser strikes each cell, scattering the light. The machine reads those scattered light angles kind of like a cellular fingerprint. A low-angle forward scatter, that tells us the cell's overall size. A higher-angle side scatter, that actually looks inside, revealing the internal complexity, the nucleus, and the refractive index. By combining these tools, the machine separates cell types with just incredible precision.
Moving on to Section 2, RBCs, indices, and histograms.
All right, so for a healthy patient, the first thing to know is this incredibly handy rapid math check we call the rule of threes. It's basically the lab's internal consistency check. You take the RBC count, multiply it by 3, and that gives you a rough estimate of the hemoglobin. Multiply that hemoglobin by 3, and you get an estimate for the hematocrit. If that mathematical relationship fails badly, that's a massive red flag. It means you're either looking at a real underlying hematological issue, like a hemoglobinopathy, or, and this happens a lot, a spurious lab error that you definitely need to investigate.
Now, those derived indices, they tell us the actual story of the cells. The MCV tells us the average size. So is the cell microcytic, normocytic, or macrocytic? Then you have the MCH and MCHC, which tell us about the hemoglobin content and concentration. Basically, is the cell hypochromic? And then there's the RDW, or red cell distribution width. This is really just the machine's numerical version of looking at anisocytosis on a peripheral smear. It tells us how wildly those cell sizes vary. But a quick heads up, high levels of monoclonal proteins can actually mess with these numbers, totally artificially, so always keep an eye out for that. To really visualize all this size variation, the machine groups the red blood cells and plots them out, giving us an RBC histogram. Imagine cell size on the x-axis and the frequency of cells on the y-axis. You usually get this nice bell curve. But you want to pay very close attention to the shoulders of that curve. If you see an abnormal bump on the left shoulder, it means you have unexpectedly small events. Think dangerous schistocytes or microspherocytes. A bump on the right shoulder, that's unexpectedly large events, which could be a massive flood of young reticulocytes or even red blood cells aggressively clumping together. Speaking of clumping, let's talk about the MCHC. A genuinely high MCHC is actually super rare because there's literally a biological limit to how much hemoglobin you can physically pack into one single cell. So if you see a high MCHC, stop and investigate. Is it real, like in hereditary spherocytosis, or is the machine being tricked? For example, cold agglutinins can make red cells stick together. The machine sees 5 stuck together cells and thinks, oh, one giant cell. That falsely drops your RBC count and artificially skyrockets the calculated MCHC. Even things like lipemia or crazy high white blood cell counts can optically interfere and falsely pump up that hemoglobin reading. Section 3. Platelet and reticulocyte analysis.
So, reticulocytes, these are the young red blood cells that just ditched their nucleus, but they still have some residual RNA hanging around inside. Modern analyzers use fluorescent dyes that specifically bind to this RNA. What does that mean? Well, more fluorescence equals a younger cell. The machine gives you a raw percentage and an absolute count, but it also calculates the immature reticulocyte fraction, or IRF. On platforms like Sysmex, this IRF bundles together the medium and high fluorescent cells. It's a fantastic, direct look at exactly how hard the bone marrow has been producing new cells over the last few days.
Now, platelets. Believe it or not, these guys can be surprisingly tricky to count accurately. Because they're just so tiny, severely microcytic red blood cells or cell fragments can easily crash the party and impersonate them during a standard impedance count. That is exactly why we often bring in optical scatter, dedicated fluorescence channels, or even high-end flow cytometry to clear through the noise and find the true platelets. We also look at the MPV, the mean platelet volume. It's a great clue. If platelets are being destroyed out in the periphery, the marrow panics and pumps out large young platelets, driving that MPV right up.
And just like with the red cells, we can measure the immature platelet fraction, or IPF. This is an estimate of those young RNA-rich platelets. A high IPF, that tells you the marrow is working overtime, pumping out fresh platelets, usually in response to them getting destroyed in the periphery. On the flip side, a low IPF with a low overall platelet count suggests the marrow itself is suppressed. It's an incredibly useful concept, but I should mention that a lack of standardization across different manufacturer brands kind of limits how universally we can apply it clinically right now.
All right, Section 4, WBCs and the differential. You've probably heard people talk about 3-part versus 5-part differentials. Here's the deal. Older 3-part analyzers use a weak lysine chemical to shrink the white blood cell membranes, and then they basically just sort them by physical volume. You get small cells like lymphs, intermediate cells like monocytes, and big ones like mature granulocytes. It's pretty broad. But modern 5- and 7-part analyzers, way better. They use multidimensional data to precisely identify all 5 specific mature cell types. The newer 7-part systems can even pull out immature granulocytes and nucleated red blood cells. How do they do it? Advanced machines hit these cells with multiple forces at the exact same time. They use direct current to figure out the total cell volume, while special lacing reagents expertly strip the cell membrane. But the real magic is high-frequency radiofrequency conductivity. The RF actually penetrates right into the cell to look at the size and density of the nucleus. Because of this, 2 cells that look totally identical in overall size on the outside can be easily told apart by their completely different internal structures.
Next, the machine takes all this rich, multidimensional data, the size, the internal complexity, the fluorescence, and plots it on a scattergram. And honestly, it's visually stunning. Normal white blood cells form these incredibly neat, predictable clusters in very specific zones. But when abnormal cells are present, say blasts or reactive lymphocytes show up, they don't follow the rules. They create weird new clusters, they overlap, or they shift completely off the normal map. When the analyzer sees that dramatic shift, boom, it immediately throws up a flag.
Section 5. Pre-analytical errors and flags. Listen, you absolutely have to know your pre-analytical errors because half the time, weird CBC results aren't actually a disease, they're just artifacts. If a sample sits around too long, the cells swell up, inflating the MCV and the MPV. If the tube is underfilled, you end up with too much EDTA, causing massive dilution and morphological distortion. And watch out for EDTA-dependent pseudothrombocytopenia. That's a mouthful, but it just means the anticoagulant makes the platelets aggressively clump together in the tube, giving you a terrifyingly low but completely fake platelet count. Oh, and if blood is drawn too close to an IV line, you'll just dilute the whole sample and artificially tank all your counts.
And I really cannot overstate this point. Analyzer flags are warnings. They are not diagnoses. If you get a flag for blasts, atypical lymphs, or giant platelets, the machine is basically saying, hey, I see a scattergram pattern or particle size I don't understand. It's a technological cry for help, and it desperately needs a human to look at a peripheral blood smear to confirm what's actually going on. Also, keep in mind, if the abnormal cells make up less than 5% of the sample, the machine's sensitivity drops, and it might miss them entirely. So a lack of a flag doesn't definitively guarantee there's zero problem.
Finally, Section 6, the clinical interpretation workflow. This 6-step approach is your absolute safeguard. It keeps you from jumping straight to a terrifying diagnosis before confirming reality. Number 1, always verify the specimen quality first. 2, review the core measured numbers. 3, assess those derived indices to see if the internal math makes sense. 4. Visually inspect the histograms and scattergrams for abnormal shifts. 5. Check all the analyzer flags. And 6. Validate anything suspicious with a manual peripheral smear. And, of course, correlate it with your actual patient. Also, a quick nod to the future. Modern analyzers are already exploring advanced cell population data to track hematopoietic progenitor cells, and even looking at early investigational sepsis parameters, which is pretty amazing.
As we wrap up this explainer, I want to leave you with one final provocative thought to ponder the next time a printout crosses your desk. The hematology analyzer is an undeniably powerful tool. We're talking about mapping cellular physics with lasers and electricity at blistering speeds. But remember, the machine doesn't recognize disease. It only recognizes particles. So ask yourself, are you treating the biological reality of the patient sitting in front of you, or are you simply reacting to the unvalidated assumptions of a particle counter?